6.3 Metabolic Pathways and Enzymes. An enzyme is a protein molecule that functions as an organic catalyst to speed up a chemical reaction without itself being affected by the reaction. In this part of the chapter, we'll also see that enzymes allow reactions to occur under mild conditions and that they regulate metabolism, including the elimination of side reactions. First, we will mention that not all enzymes are proteins. Ribozymes, which are made of RNA instead of proteins, can also serve as biological catalysts. Ribosomes are involved in the synthesis of RNA and the synthesis of proteins at the ribosomes. Let's also recognize that reactions do not occur haphazardly in cells. They are usually part of a metabolic pathway, which is a series of linked reactions. Metabolic pathways begin with a particular reactant and terminate with an end product. While it is possible to write an overall equation for a pathway as if the beginning reactant went to the end product in one step, actually many specific steps occur in between. In the pathway, one reaction leads to the next reaction, which leads to the next reaction, and so forth in an organized, highly structured manner. This arrangement makes it possible for one pathway to lead to several others, because various pathways have several molecules in common. Also, metabolic energy is captured and used more easily if it is released in small increments rather than all at once. A metabolic pathway can be represented by the following diagram. A, enzyme 1 turns this into B. B, with the use of enzyme 2, turns it into C. C, with the enzyme number 3, turns into D. D, using enzyme 4, turns into E. E, using enzyme 5, turns into F. And then F, using enzyme 6, turns into G. In this diagram, the letters A through F are reactants, and the letters B through G are products in the various reactions. In other words, the products from the previous reaction become the reactants of the next reaction. The letters E1 through E6 are enzymes. Any one of the molecules A through G in this linear pathway could also be a reactant in another pathway. A diagram showing all the possibilities would be highly branched. The energy of activation. Molecules frequently do not react with one another unless they are activated in some way. In the lab, for example, in the absence of an enzyme, activation is very often achieved by heat and reaction flask to increase the number of effective collisions between molecules. The energy that must be added to cause molecules to react with one another is called the energy of activation, EA. Figure 6.5 compares the EA when an enzyme is not present to when an enzyme is present, illustrating that enzymes lower the amount of energy required for activation to occur. The energy content of the product remains the same, however. Enzymes allow reactions to occur under mild conditions by bringing reactants into contact with one another and even by participating in the reaction at times. Let's look at figure 6.5. So in this diagram, we see the progress of the reaction on the x-axis, and we see the free energy on the y-axis. In red and blue on the left side of the chart here, we see the energy of the reactants. The energy of activation on the blue chart is much higher than the energy of activation on the red line of the chart, which is the energy of activation with the enzyme. So in this situation, the product's energy at the very right side is still the same, and the energy of the reactants on the very left side is still the same. It's just how much energy is required to make the reaction occur. And with enzymes, it is less energy required. Let's keep reading. The enzyme-substrate complex. The reactants in an enzymatic reaction are called the substrates for the enzyme. Considering the metabolic pathway shown previously, A is the substrate for the enzyme 1, and B is the product. Now B becomes the substrate for enzyme 2, and C is the product. This process continues until the final product G forms. The following equation, which is pictorially shown as figure 6.1, is often used to indicate that an enzyme forms a complex with its substrate. So the enzyme plus the substrate yields the enzyme-substrate complex, which then releases the enzyme to be reused again, and the product is made. In most instances, only one small part of the enzyme called the active site binds with the substrates. It is here that the enzyme and substrate fit together, seemingly like a key fits into a lock. However, it is now known that the active site undergoes a slight change in shape to accommodate the substrates. This is called the induced fit model, because the enzymes is induced to undergo a slight alteration to achieve optimum fit. This induced fit model is very important for the AP exam, as well as knowing what the active site is and how these substrates fit specifically into the enzyme. Let's keep reading. The change in shape of the active site facilitates the reaction that now occurs. After the reaction has been completed, the product is released, and the active site returns to its original state, which is ready to bind to another substrate molecule. Only a small amount of enzyme is actually needed in the cell because enzymes are not used up by the reaction. Some enzymes do more than simply complex with their substrates. They participate in the reaction. Trypsin digests protein by breaking peptide bonds. The active site of trypsin contains 3 amino acids with R groups that they actually interact with the members of the peptide bond. First to break the bond and then to introduce the components of water. This illustrates that the formation of the enzyme-substrate complex is very important in speeding the reaction because enzymes only complex with their substrates. They are sometimes named for their substrates and usually end in ase. For example, lipase is involved in hydrolyzing lipids, so we call it lipase. So let's look at this figure 6.6. So in part A of this figure, we see the substrates in yellow. The substrates fit perfectly into the active site of the enzyme, turning it into the enzyme-substrate complex. This, in turn, allows the substrate to undergo the reaction that creates the products, which are green and pink. The enzyme itself is left alone and ready to react with another substrate. In the situation in part B, the 2 substrates are green and pink, and they fit perfectly into the active site of the enzyme, which is orange. It is now called the enzyme-substrate complex. Now, the 2 substrates that were green and pink, they react with each other and create the yellow product. It is released, and the enzyme itself is left alone and ready to combine with more substrates to create more products. Let's keep reading. Regulation of metabolism. Because enzymes are specific, they participate in regulating metabolism. First, which metabolic pathways are being utilized is dependent on which enzymes are present. Since metabolic pathways can intersect, the presence of particular enzymes can determine the direction of metabolism. Then, too, some particular reactants can produce more than one type of product. Therefore, which enzyme is present determines which product is produced and the direction of the metabolism without several side pathways being activated. Factors affecting enzymatic speed. Generally, enzymes work quickly, and in some instances they can increase the reaction rate more than 10 million times. The rate of the reaction is the amount of product produced per unit of time. The amount of product per unit of time depends on how much substrate is at the active sites of enzymes. Therefore, increasing the amount of substrate and also the amount of enzyme can increase the rate of reaction. Any factor that alters the shape of the active site, such as pH or temperature or an inhibitor, can decrease the rate of reaction. Finally, some enzymes require cofactors that help speed the rate of reaction because they help bind the substrate to the active site or they participate in the reaction at the active site. The substrate concentration. Molecules must collide to react. Generally, enzyme activity increases as substrate concentration increases because there are more collisions between the substrate molecules and the enzymes. So, to reiterate, if I increase the amount of substrate, I create more interactions, and so it can speed up the reaction. As more substrate molecules fill the active sites, more product results per unit of time. But when the active sites are filled almost continuously with substrate, the rate of reaction cannot increase anymore. Maximum rate has been achieved. Just as the amount of substrate can increase or limit the rate of the enzyme reaction, so the amount of active enzyme can also increase or limit the rate of the enzymatic reaction. Basically, if you fill all of the active sites with the substrate, it can't go any faster. All of the active sites are full. So there's also optimal pH. So each enzyme also has an optimal pH at which the rate of reaction is the highest. Figure 6.7 shows the optimal pH for the enzymes pepsin and trypsin. At this pH value, these enzymes have their normal configurations. The globular structure of an enzyme is dependent on interactions such as hydrogen bonding between the R groups. A change in the pH can alter the ionization of these side chains and disrupt normal interactions. And under extreme conditions of pH, the enzyme becomes inactive. Inactivity occurs because the enzyme has an altered shape and is then unable to combine efficiently with its substrate. So look at this chart. It shows an increasing in pH from 0 to 12. Pepsin works most effectively at about a pH 2.3, but trypsin works a little bit more effectively at a pH of about 8. So pepsin is found in the acidity of your stomach, while trypsin is found more in your small intestine. Okay. Now let's look at temperature. Temperature, typically as the temperature rises, enzyme activity increases. If we look at figure 6.8 right here, so we see the increase of temperature from 0 to about 45 and you reach the optimal temperature. After that, you start denaturing the protein or the enzyme, and so it's not as effective past 60 degrees. So a lot of times in digestion, certain animals like these iguanas here, they need to go find a warm rock to help their enzymes react at the most effective temperature for their digestion. Other animals like polar bears, which are endothermic like us, we actually can keep our body temperature at the correct, most efficient temperature. This occurs because warmer temperatures cause more effective collisions between the enzyme and the substrate. The body temperature of an animal seems to affect whether it is normally active or inactive. It has been suggested that mammals are more prevalent today than reptiles because they maintain a warm internal temperature that allows their enzymes to work at a rapid rate. In the laboratory, if the temperature rises beyond a certain point, enzyme activity eventually levels out and then declines rapidly because the enzyme is denatured. An enzyme shape changes during denaturation, and then it can no longer bind to its substrates efficiently. Exceptions to this generalization do occur. For example, some prokaryotes can live in hot springs because their enzymes do not denature. These organisms are responsible for the brilliant colors of the hot springs. Another exception involves the coat color of animals. Siamese cats have inherited a mutation that causes an enzyme to be active only at cooler body temperatures. Their activity causes the cooler regions of the body, the face, the ears, and legs and tail, to be dark in color. The coat color pattern in several other animals can be explained similarly. For example, in this Siamese cat, they've inherited a mutation that causes an enzyme to be active only in the cooler body temperatures. Therefore, only certain regions of the body are dark in color. So let's keep reading about enzyme cofactors. Many enzymes require the presence of an inorganic ion or non-protein organic molecule at the active site in order to be active. These necessary ions or molecules are called cofactors. The inorganic ions are metals such as copper, zinc, or iron. The non-protein organic molecules are called coenzymes. These cofactors participate in the reaction and may even accept or contribute atoms to the reactions. In the next section, we will discuss two coenzymes that play significant roles in photosynthesis and cellular respiration, respectively. Vitamins are relatively small organic molecules that are required in trace amounts in our diet and in the diets of other animals or synthesis of coenzymes. The vitamin becomes part of a coenzyme's molecular structure. If a vitamin is not available, enzymatic activity will decrease, and the result will be a vitamin deficiency disorder. Niacin deficiency results in a skin disease called pellagra, and riboflavin deficiency results in cracks at the corners of the mouth. Looking at figure 610, cofactors at active site. Cofactors, including inorganic ions and organic coenzymes, may participate in the reaction of the active site.